Hello everyone, can you see me? Can you hear me? Give me a minute to confirm. If I'm live, I will start the session ahead. Okay, yeah, I think it's working. So can you give me a thumbs up if video, audio, everything is open? Just a second. Everything is fine. Is everything fine? Vishwas, Shubhangi, Dopamine, Osama, Rohit, all others. Give me a thumbs up, anyone. Okay. So Vishwas, is everything fine? Can you see me clearly? Can you hear me clearly? Can you see the PPT also? Is everything smooth? Can I start the session? So I guess it's working. Okay. So very good evening to all of you. I welcome you all for today's session. I am Dr. Priyanka Sachdev here, and today I'm here to take a very important topic on huge demand of students, that is from microbiology, general microbiology, that is bacterial genetics. Now, many students find this topic very, very difficult. That's why I decided to schedule a session on this, and many students requested me personally to take this topic in detail. Now, this topic is very important for all competitive exams as multiple questions, MCQs comes from this topic. Many image-based questions are also created from this topic, and also this topic is going to be super duper important for second Prof MBBS students who are due for their university exams of microbiology. So this question is going to become in long question as well as in a part as a short question. So I guess without wasting any further time, I must start the session ahead, right? Okay. So I am teaching you bacterial genetics. Before understanding bacterial genetics, you must understand the structure of the bacterial DNA. Now, the bacterial DNA is a circular chromosome with double-stranded DNA. You can see it's a double-stranded DNA and it's in the form of a circle. That is the structure of a bacterial DNA. Now, the bacterial DNA, this is the chromosome. You know, it is a chromosome. The chromosome is single and it's circular. You know, it's a circular single chromosome in the bacteria. So this is, the chromosome is made up of DNA, right? Of course, the chromosome made up of DNA, we all know. Do you know any extra chromosomal genetic material present in the bacteria? Extra chromosomal. It should not be a part of chromosome, but it should be DNA because it is genetic. So tell me any part in the bacterial cell which is DNA but which is not a chromosome. That is extra chromosomal genetic material. Do you know? It's a very, very important MCQ, right? Now, extra chromosomal genetic elements are two. In bacterial cells we are talking, not in human cells. So they are plasmids and episome. Both of them are DNA, but they are not chromosomes. They are DNA, but they are not chromosome. I'm repeating again. So that's why they are known as extra chromosomal genetic element. Yes, Shubhangi, absolutely right. So one is plasmid, one is episome. Now, what do you mean by plasmid and episome? What is the difference between the two? Now, if the DNA is present in cytoplasm, not in nucleus, if the DNA is present in cytoplasm, it is known as plasmid. It is not a chromosome. And if the DNA is present in nucleus, it is integrated with the chromosome but not a chromosome. So it is known as episome. Here you can see both, right? Can you see this plasmid here? It is present in the cytoplasm. It is present in the cytoplasm. It is DNA. It is genetic material. It is DNA in nature, but it is not a chromosome. That's why it is known as plasmid. And if this plasmid goes here and integrate with the chromosome, it is not a chromosome. It is integrating with the chromosome. So it is present in the nucleus. So the same plasmid will be known as episome. Give me a thumbs up everyone on this point if you got the difference between the plasmid and the episome. Now a beautiful diagram in front of you to explain this concept. Listen, everyone listen here. Can you see here? This is a green color bacterial cell. Can you all see? This is a bacterial cell. In the bacterial cell, please concentrate the DNA, the chromosome of the bacteria. The red color I'm marking right now. This is the bacterial DNA, or you can say bacterial chromosome to be simplified, right? Now, in this diagram, can you all see what are these circular structures? These circular structures. These circular structures are present outside the nucleus. They are present in cytoplasm. Basically, they are DNA in nature, but they are in cytoplasm. That's why known as plasmid. Yes, they are plasmid, right? Now, when the bacterial cell, the parent cell is dividing into two daughter cells, you know, bacterial replication taking place from one parent cell, two daughters are formed. So, the DNA divides, we know. Can you see this DNA is dividing, the chromosome is dividing, but the extra chromosomal DNA is also dividing. That is, I mean to say, plasmids replicate. Like chromosome only, plasmids replicate in the same way. Plasmids are replicable in the same way as nucleus is replicating. You got my point? In both the daughter cell, the same number, exactly same plasmids are there as compared to parent cell. So they are doubling, they are replicating. We got it from this diagram. Now, in the second diagram below, See the below diagram, the lower half. You can see here, this is a plasmid. It is going in the nucleus and integrating. Now in this cell here, please everyone concentrate here. Here you can see the red one is the chromosome, but the blue one is not chromosome. It is DNA. It is the plasmid which is integrated with the nucleus. Now it is not known as plasmid. Now it is known as episome. Now it is known as episome, and episome is also replicating in the same way as plasmid. So what we have learned, the summary is that there are two types of extra-chromosomal genetic material. It is known as plasmid and episome. When it is present in cytoplasm, it is plasmid. When it is present in nucleus integrated with the chromosome, it is known as episome. And plasmid, episome, both are replicable in the same way as bacterial chromosomes do. Give me a thumbs up on this point, on this summary. Everyone. So have you got it? Can we proceed ahead? You got it? So what we have learned plasmid and episome both replicate independently in the same way as the bacterial chromosome is doing. Right? So that is the thing. Now let's come on gene transfer. This is a bacterial cell. Let me draw a bacteria in front of you. This is a bacterial cell. Okay. This is the circular DNA. This is the circular DNA chromosome of the bacteria. Right? This is another bacteria. Let me draw another bacteria. This is bacteria number 2, and this is the DNA of this bacteria. Is it possible, I'm asking you, is it possible to transfer the genes of this bacteria to this or this bacteria to this, vice versa? The transfer of genes I'm asking. The transfer of the genetic material, the transfer of the genes, the transfer of the DNA. What are the ways one bacteria can transfer the genes to another bacteria? What are the ways? So we are having 4 ways. Yes, of course it's possible, Shadab. Absolutely right. It's possible. So there are 4 ways by which we can do it. Of course. So what are the 4 ways by which bacterial material, the genetic material can be transferred from one bacteria to another? So the 4 ways are transformation, transduction, lysogenic conversion, and conjugation. So this is today's topic. You have to understand the difference between the 4, the importance, the pros and cons of each method, right? So these are the various methods by which the bacteria can transfer gene from one bacteria to another, right? Now let me discuss these one by one in detail. In the end, we will solve some MCQs based on that, right? So let me start with the first method, transformation. Transformation. The word transformation you have to learn is the naked DNA. What do you mean by the term naked? Okay. I will draw a diagram for you, right? Imagine this is a bacteria. Okay, this is a bacteria you all can see. This is the DNA of this bacteria. By DNA, I mean the chromosome of the bacteria. This bacteria is dead. What I am saying? This bacteria is dead. Once this bacteria is dead, the bacteria will rupture. The bacteria will rupture. So this is the rupture of this bacteria. When the bacteria will rupture, the chromosome will leak out. This red color chromosome, it is leaking out. Now it is free. Now it is free DNA. Or else, in other words, I will say it is naked DNA. It is naked DNA. Free or naked DNA is present. Because it is a DNA present inside the bacteria is already dead. Now this DNA is free DNA. Now let me draw another bacteria. This is another bacteria. Absolutely different bacteria, and this is the chromosome of this bacteria, particularly this bacteria. Now this naked DNA, the red one, which was from the first bacteria, this directly entering the second bacteria without any vehicle, without any agency. Directly वो खुद ही enter कर रहा है, उसको किसी agency की जरूरत नहीं है, किसी transporter, किसी vehicle की जरूरत नहीं है. So the naked DNA is entering inside another bacteria and integrating with the DNA of that bacteria. So blue and red integration is taking place. Blue is the DNA of this bacteria and red is the DNA which is coming from the dead bacteria. So this method is known as transformation. In transformation, basically, you have to learn a word. It's naked DNA, naked. And from where the term naked is coming, it's the dead bacteria. The bacteria is dead, from which the DNA is leaking out and entering into another. Everyone give me a thumbs up, fatafat. Give me a thumbs up, come on. Are you people there? Shubhangi, Purnima, Anilipsa, Vishwas, Osama. Give me a thumbs up, come on. So, are you guys there? Can we move ahead? So, transformation is naked. So, you people believe me, but you shouldn't. You should believe science. You should ask me, ma'am, give me proof that this transformation happens. Why we should believe you? You are saying that this happens, but we are not agree. So, do you have some experimental proof to prove this method? So, yes, we have a great scientist who has proved this method. The method, I'm talking the first method, transformation, in which the gene transfer takes place from one bacteria to another through naked DNA. Naked. The first bacteria is dead. So it releases or leaking out the free DNA. The free DNA from the dead bacteria, it is coming in the environment, and from the free environment, it is entering the second bacteria. After entering, it is integrating with the DNA of that particular live bacteria. So this method is known as transformation. Now, we were talking about the proof. What is the proof? So the great scientist is the Griffith. Griffith has already proven it, right? So he has done a beautiful experiment. A beautiful experiment. I will explain you what is the experiment. In the end, you will also convince, yes ma'am, transformation takes place. So you will convince in the end. Now you have learned the definition. Now let me do an experiment in front of you. Let me explain you the experiment so that you get convinced that yes, transformation basically takes place. Okay, we will do that experiment. So the Griffith has done an experiment on a bacteria. The name of the bacteria is Pneumococcus. Pneumococcus is a bacteria. Okay. Pneumococcus are of two types. Let me draw the two types of pneumococcus in front of you to explain. Okay. So this is the first pneumococcus. This is the second one. There is one difference between the two. The first one has a capsule. The second one do not have a capsule. You got it? So there are two types of pneumococcus in the world. Two types of pneumococcus. The capsulated one. You can see capsulated one. So since the capsule is forming a covering, it is known as smooth one. It is known as smooth one. It is smooth from the above. And the second one is non-capsulated. It doesn't have any capsule above it. So that's why it is rough. It is very rough in appearance. It is not smooth. It doesn't have any covering. This one is known as rough form. So smooth pneumococcus and rough pneumococcus. That is capsulated and non-capsulated. Yes, I am coming on that point, Shubhangi. So Shubhangi is absolutely right. Capsule is necessary for producing a disease, for producing virulence. If you want pneumococcus cause disease in any animal or human, it should have a capsule. Without capsule, pneumococcus do not cause any disease. They are harmless. You got my point? So I mean to say the capsulated form are virulent and the non-capsulated form are avirulent or non-virulent or avirulent. You can see. Okay? These are non-virulent one. Non-virulent one, these are virulent. You got it? So if I say smooth one, you must understand it is the virulent one, harmful, dangerous. They can cause disease, they can cause death. But the rough form are avirulent. They doesn't cause any disease. Give me a thumbs up on this point. Give me a thumbs up on this point. So Shadab, you got it? The virulent one are the capsulated one or the smooth one. So capsulated, smooth and virulent. These three things are same. Non-capsulated, rough and avirulent. These three things are same. I guess you got it. Okay. Now we will do an experiment. So Griffith has taken 4 mice, 4 animals, 4 live mice. So mice number 1, 2, 3, 4, and he has done an experiment. Let me do this experiment in front of you. Explain you. So this is mice number 1. This is mice number 2. This is mice number 3. And this is mice number 4. Imagine you are doing this experiment. In the first mice, oh God, I have to draw it again. In the first mice, I'm sorry, he has injected, let me draw it. 1, 2, 3, 4. In the first mice, Griffith have injected capsulated one. Capsulated one means smooth. Smooth live bacteria. You have to tell me the result. What will happen to this mice? Smooth one and the live. Right? In the second, he has injected the rough one, that is non-capsulated. Rough one and live bacteria. In the third mice, he has injected again smooth one. Again smooth one, that is again capsulated, the virulent one, but this time not live, the dead bacteria. And in the fourth, he has mixtured second and third. He has taken half this and injected both in the fourth one. Right? You have to tell me the results in each of them. I will explain you. So in the fourth one, he has done 2 plus 3. 2 plus 3. The combination of 2 plus 3 is equal to 4. So in 2, he has taken rough and live along with smooth and dead. Smooth and dead. Now you have to, that is this one and this one. Right? So this combination he has taken. Now apply the knowledge what I have given you. I have told you smooth one are virulent. So what will be the result of this mice? You say, ma'am, this mice will be dead because it will cause disease. And the bacteria are live. They are virulent. They are live because they are capsulated. So this bacteria will be dead in the end. Give me a thumbs up, everyone. Everyone, this bacteria will be dead. Common sense. You will say, yes, ma'am, we can understand it. What will be the result in the second mice? In the second mice, he have injected rough one. You will say, ma'am, rough one are harmless. They do not cause any disease. Come on, right? So because they are non-capsulated, although they are live, but they are harmless. Right? They don't have any capsule. So this mice will be live and healthy. I must write the word healthy. Healthy in the end. No disease, nothing. Right? What about the third mice? Can you tell me what will happen? Osama, Purnima, Shubhangi, what will happen in the third mice? Can you tell me? This mice is again smooth. You will say, ma'am, smooth one are dangerous. They can cause disease, they can cause death. But the bacteria are dead. Since they are dead, they cannot cause the disease. They cannot multiply. After going inside the mice, they cannot multiply because they are already dead. Although they are capsulated, I agree. The capsulated one are virulent, but they are dead now. Since they are dead, these one are live. So this mice will also be healthy. Yes, this mice, no, Shubhangi. The third one live. Yeah, okay, fourth one you are saying. Okay, the third one will also be healthy and live. Right. Now, see the result of the second and third. In the second, it was healthy. In the third, it was healthy. So, common sense. If you use common sense, if I ask you a question, if you mix second and third and inject in the fourth one, so we are expecting it should be healthy, because when we are giving individually second one, it is healthy. Individually third one, it is healthy. But when we are mixing second and third now, we are expecting it should be double healthy. It should be healthy. But on the contrary, this mice is dead. Now, this is surprise. It was a surprise. Why it is happening? Why it is happening? Everyone was shocked. Why it happened? Now, none of the two can cause disease. None of the two can cause death. Listen, if we talk about rough life individually, they cannot cause disease. If we talk about smooth dead individually, they cannot cause disease. But yeah, smooth live can cause disease. Smooth live, but we have not injected smooth live, come on. So why this mice is dead? Why this mice? Yes, the answer is transformation. Very right, very right. So basically, what was transformation? The bacteria which is dead is lysed, is ruptured, and the DNA from that bacteria will come out and enter in another live bacteria, right? So basically here smooth one are dead. The smooth one, the capsulated one are dead. So they are ruptured. So from them the DNA is coming out and the DNA is entering into the live rough one. And this DNA contains the gene for the capsule. So rough one will produce capsule because now it has the gene for capsule. It will start forming capsule. Once it will start forming capsule, it will cause the death of the mice. So basically this is happening inside the fourth mice. Got it? Give me a thumbs up if you got it. Everyone. So this is Griffith experiment. Anyone having any doubt? If you don't have doubt, jitna audience utna thumbs up. Everyone give me a thumbs up. Come on. So this is Griffith experiment. You have to explain in your own words, in your own terms, right? What is the result in the 4 mice? Beautiful diagram explaining the same thing. See what we are injecting in the first mice, see the result. What we are injecting in the second, see the result. What we are injecting in the third, see the result. We are combining second and third and injecting in the fourth one, see the result. So the fourth mice is the dead, explaining the transformation. So transformation is explained in this mice, the fourth one. The fourth one is explaining the transformation. I guess everyone got it. Everyone, right? So this was the first method. The method is known as transformation. That is the DNA or the genes from one bacteria is coming out and entering in another bacteria through naked DNA. No agency, no vehicle. That is transformation, and you have to explain Griffith experiment. So Griffith experiment is on a bacteria that is Pneumococcus. It is an MCQ. And it is for explanation of transformation. It is an MCQ. And you should know the details of the Griffith experiment for second Prof university exams. Let me proceed to the next method, that is transduction. Give me a thumbs up. Are you people with me? Can I go ahead? Okay. Transduction. Now, here what will happen now? You know, there is a virus which can infect bacteria. The virus can infect humans also. We have viral infections. Humans can have. In the same way, virus can infect bacteria also. Such a virus which infect bacteria is known as bacteriophage. The first concept, bacteriophage is not a bacteria. Bacteriophage is a virus which infect bacteria. What I am saying? Repeat again. Bacteriophage is a virus which infect bacteria. Right? Now imagine, let me draw the diagram for you. This is a bacteria. Right? Inside this bacteria, this is the DNA of this bacteria. Now there is a bacteriophage coming. This is a bacteriophage. It is a virus coming. So this virus, it is the DNA of this virus. It is entering inside this bacteria. It is infecting. So the virus is inside the bacteria. The red color DNA is the DNA of the bacteria and green color DNA is the DNA of the virus. So this virus will multiply inside the bacteria, forming multiple progeny using the machinery of the bacteria. But during this, when from one virus, multiple viral progenies are formed inside bacterial cell. So during this process, some packaging error take place. What take place? Packaging error. What do you mean by packaging error? Normally, when the virus is replicating now, so all the DNA in all the progeny should be of virus only. But a little bit amount of this bacterial bacterial DNA can enter in the viral progeny. A little bit, not much, but a little bit in confusion. This is known as packaging error. Now, when these viruses come out, the progeny, the bacteria will burst and this progeny will come out, right? Now, this progeny have a small amount of DNA which is from the bacteria, the red one. Now, this bacteria, this virus will go to another bacteria. Let me draw another bacteria, bacteria number 2. Another bacteria, this is the DNA of this bacteria. This virus will go to this bacteria and give the red DNA which he has taken from that bacteria to this bacteria. So basically, the DNA is transferred from one bacteria to another through a vehicle. The vehicle here is the virus. The vehicle here is the bacteriophage. So agency is the bacteriophage. So basically, a virus is coming. It is infecting one bacteria, taking a small amount of DNA from that bacteria, then it is coming out and entering another bacteria, giving the DNA which he has taken from that bacteria to this bacteria. So the DNA is transferred from one bacteria to another bacteria via virus. Give me a thumbs up. This is known as transduction. Transduction is through bacteriophage. You got it? You got it? Everyone got it? So you have to learn one word here, packaging error. This is due to packaging error. Explain in your own words if you are a university exam candidate. Otherwise, you can learn it. Yes. So you can learn it and for the MCQs. So you can see the same thing here. See, this is the first bacteria. See, this is bacteriophage entering the first bacteria. This is the DNA of that bacteria. So multiple progenies are formed, but packaging error take place. So this progeny is coming out and after that it is infecting another bacteria. So this is the same story. You got it? You got it? So this is transduction. In transduction, who is the vehicle? The vehicle is a virus, that is bacteriophage. Transduction is very, very important tool, excellent tool for genetic engineering, the best tool, right? And among the bacterial various methods, this is the most widespread mechanism. So we are done with transformation and we are done with transduction. In transformation, no vehicle was involved. Naked DNA was transferring from one bacteria to another directly without any agency. But in the second method, that is in transduction, transfer take place via an agency. The agency or the vehicle is a bacteriophage, which is a virus. Let me come on the third method. The third method is lysogenic conversion. Give me a thumbs up. Are you people there? Can I continue? So the third method is the lysogenic conversion. So actually in lysogenic conversion also I want to explain you two things. Again, here also bacteriophage will be involved. Then you will ask, ma'am, what is the difference between transduction and lysogenic conversion? I will explain you in the end. Okay, don't worry. So first understand what is lysogenic conversion. In lysogenic conversion, I want to explain you two things. What two things? Virulent or lytic cycle and temperate or non-lytic cycle. Now many MCQs come from this point. You will say, ma'am, what does it mean? So don't worry, I am explaining you. Listen, again we will start the same story. This is bacteria. You can see this is the bacteria. Can you see? This is a bacteria inside which the virus is entering. This is a virus. The virus is the bacteriophage. It is entering inside the bacteria. You can see the bacterial DNA is shown by blue color and the viral DNA is shown by red color. After entering, there are two possibilities. Either the DNA of the two merge with each other. Red, blue, red, blue, red, blue. So merging, integration. This is known as integration. The two DNA merge with each other. The DNA of the bacteriophage merges with the DNA of the bacteria. This is known as integration. Integration is non-lytic. It is known as temperate cycle. Temperate means non-lytic. The second possibility is that the DNA are not merging with each other. DNA of bacteria is blue only and DNA of virus is red only. But the only thing, virus is replicating inside the bacteria. The virus is replicating inside the bacteria, forming multiple colonies. In the end, the bacteria got ruptured and multiple progenies are coming out. This is known as simply replication. Replication is the lytic cycle. Right? Here I am interested in the second one, that is integrate cycle. Integration mein what is happening? The two DNA are merging with each other. So that's known as lysogenic cycle. You got my point? You will say, ma'am, then what is the difference between transduction and here in lysogenic conversion? In transduction, actually, listen, the bacteriophage was acting as a vehicle. It was just a vehicle. It was only transmitting the DNA from one bacteria to another. Here actually the DNA of the bacteriophage is merging with the bacteria. You got my point? Let me explain you. Don't worry. Don't get confused. So this is bacteria number 1. This is bacteria number 2. Here it is having red color DNA. Here it is having blue color DNA, right? Let me draw a phage. This is a bacteriophage having purple DNA. Now see, I have drawn 3 colors to explain you something. The red DNA, the blue DNA, the purple DNA. This is bacteria 1. Please understand, huh? This is bacteria 1. This is bacteria 2. And this is the virus. You can see the color coding. You can use any color, right? Now the virus is entering inside the first bacteria. Due to packaging error, it is taking a little bit of DNA inside it. And this virus then going to second bacteria and transmitting the red DNA from that bacteria to this. So this virus is acting only an agency. It is only agency, taking here and dropping here. It is like just like Swiggy, taking from one place to and dropping at other. Nothing else. It is an agency. So this is known as transduction. It is only acting as an agency, a delivery boy, taking the material from one place and dropping it at another place. His own material is not involved. The viral own DNA is not involved. That is transduction. You got it? Now let me draw another diagram. Let me draw another diagram to explain you lysogenic conversion. In lysogenic conversion, we don't require 2 bacteria. We require only 1 bacteria. So not bacteria 1, bacteria 2. Only 1 bacteria having the red DNA. The red DNA. Now the virus I am drawing, this is the virus having the purple DNA. Here the virus will enter inside this bacteria and merging of the 2 DNA take place. Integration of the 2 DNA, the purple and the red. Here second bacteria is not involved. So the viral DNA is integrating with the bacterial DNA. In this way gene transfer take place. So this method is known as lysogenic conversion. Lysogenic conversion. I guess I am crystal clear in my concepts. Right. So in the second method, the virus is not acting as an agency. The viral own genetic material is involved in the gene transfer. Give me a thumbs up. I tried hard. Actually, I tried hard. So please appreciate my efforts if you got it. The difference between the two, the transduction and lysogenic conversion. In both of them, bacteriophage is involved. I agree. But there is a big difference between the two. Come on, give me a thumbs up. Come on. So only 3 students got it or everyone got it? If everyone got it, please everyone give me a thumbs up. You got it? Can we continue? So this is the lysogenic conversion. So the phage DNA will integrate with the bacterial chromosome and it will multiply synchronously whenever the bacteria is dividing. It is known as lysogenic conversion or temperate cycle, whatever you say, one and the same thing, right? So the best example is the diphtheria. The best example here is the diphtheria. You know, let me draw a bacteria. This is the diphtheria bacteria. This is the DNA of the diphtheria. Diphtheria are of 2 types. Like pneumococcus were of 2 types. Now, diphtheria are also of 2 types. Some diphtheria produce toxin. The one bacteria, the one diphtheria, it is diphtheria. The one Corynebacterium diphtheriae which produce toxin, it is virulent. Right? And the one which do not produce toxin, it is avirulent. It is avirulent. So like pneumococcus, their capsule was required for the virulence. In diphtheria, toxin is required for virulence. So who will decide that diphtheria will produce toxin or will not produce toxin? Who will decide? So for toxin production, basically the diphtheria have a gene. The name of the gene is tox gene. So the diphtheria which is having tox gene will produce toxin and it will be virulent. The diphtheria which do not have tox gene, if the tox gene is absent, it is absent. So it will not produce toxin and it is avirulent. Give me a thumbs up. This is the basic concept. There are 2 types of diphtheria. The one which produce toxin, the one which do not produce toxin. The one which produce toxin are virulent. They cause disease. The one which do not produce toxin are avirulent. They do not cause disease. So who decides whether the diphtheria produce toxin or do not produce toxin? It is the tox gene. If tox gene is present, it will produce toxin. If tox gene is absent, it will not produce toxin. So can we convert, my question to you, can we convert avirulent diphtheria to virulent diphtheria? Can we convert? So answer is yes, we can convert. If we can convert non-toxic strains to toxigenic strains, we can do so. So how we can convert non-toxic strains of the diphtheria to toxigenic strains? How can we do so? If we can transfer the tox gene. So if the non-toxigenic strain get the tox gene from somewhere, it will start producing toxin and avirulent form can be converted to virulent form. So we can do so by lysogenic conversion. So let me draw a diphtheria again in front of you. This is avirulent. This is non-toxigenic currently. Non-toxigenic because it doesn't have tox gene. It is non-toxic, non-toxigenic. So let me draw a phage. This is the phage. This is a bacteriophage inside which the tox gene is present. It contains tox gene and the best bacteriophage is beta phage. The name of this phage is beta phage. It is very important MCQ. So it is entering inside and giving the tox gene to the bacteria. Now from non-toxigenic it will convert into toxigenic and start producing toxin. So this is the best example here. Yes, absolutely right. Give me a thumbs up. You got it? So what we have learned till now, we have learned three methods. What was the first one? Transformation, in which the gene transfer takes place through naked DNA. The second one is the transduction, in which the phage, bacteriophage, acts only a vehicle, a delivery boy, taking the DNA from one bacteria and releasing it to another bacteria. That's it. But the third one is lysogenic conversion, in which the phage DNA itself acts as a genetic element for the bacteria. They are integrating the two genetic elements. One is bacterial, one is viral. Give me a thumbs up. Come on. Can we go ahead? Can we go ahead? And the last method here is the conjugation. The most important and difficult to understand is conjugation. Can we go ahead? So conjugation is simply known as, it is sex in bacteria. It is the sex in bacteria. So you can see there are two bacterias. In the first bacteria, okay, let me draw a diagram for you. This is the first bacteria. Okay, this is the second bacteria. The first bacteria, this is the nucleus. The second bacteria, this is the nucleus. But first bacteria contain a plasmid also. Second bacteria doesn't have that plasmid. So the bacteria which has plasmid, this plasmid is known as F-plasmid. The name of this plasmid, you know what is a plasmid? I have already explained you. Plasmid is extra-chromosomal DNA present in the cytoplasm. So the first bacteria have F-plasmid. It is known as male bacteria. Male bacteria, F is for fertility. It is having the fertility plasmid, that's why known as male bacteria. In which the second bacteria in which F plasmid is absent, it is known as female bacteria. It is known as female bacteria. So what will happen between them? A tube is formed. It is known as conjugation tube. A conjugation tube or pili will form. A conjugation tube will form. So the first bacteria which is having the F plasmid, it will replicate the plasmid. It will form two copies. One for the himself, and the second copy will be transferred to the second bacteria. Now the second bacteria also have F plasmid after conjugation. So this is the transfer of genetic material between two bacteria by physical contact. That's why it is known as sex in bacteria, right? So the two bacteria are coming closer, a tube is formed between the two bacteria, and one bacteria giving the DNA material through that tube to another bacteria. So this is through physical contact between two bacteria by forming a pili or a sex tube between them. Give me a thumbs up. Now the point is that initially, before conjugation, this female bacteria was there because the plasmid is absent, but after conjugation, the female converted into male. Because it also have the F plasmid. So female converted into male. At the end of the conjugation, both are males. When they started, one was male, one was female. Give me a thumbs up if you got it. Come on. So this is conjugation. Conjugation is mating in bacteria or physical contact in bacteria. So it is the transfer of genetic material from one bacteria to another. The one bacteria which gives the genetic material is a male or a donor. The second bacteria which receives is a recipient or a female. So the same story is given here. At the start, you can see, if you can zoom this diagram, this one is the donor, male, this one is the recipient, female, right? But at the end, both are males. Both are donors now. They can be donor. And between them a sex pilus is formed. You got my point? Give me a thumbs up if you got it. So this is known as conjugation. Basically in conjugation, the definition of conjugation is physical contact in the bacteria. Between two bacteria, there is a physical contact and the genetic material is transferred from one bacteria to another through a tube. That is known as sex pilus, sex pili, a sex tube. And the genetic material is transferred from one to another. The one which gives is a donor. The one which receives is a recipient. So this is how it takes place. So it is through physical contact, and it is of three types. The first type I have already explained you, F factor or fertility factor or sex factor. So the one is male, one is female, we have already understood, right? The donors are the male and the recipients are the female. The males are F positive and the females are F negative. At the end, both will be F positive. We have already seen. So F plasmid codes for fertility, and we have understood this diagram. I guess everyone knows. So can you see the first bacteria? This is F factor present. Now it will replicate and give one copy to the second bacteria. So at the end, both are F positive. Give me a thumbs up. Come on. The second type of conjugation. Is call factor. This one was F factor. ये F factor था. जो दूसरा है ना वो call factor है. Call is not very important. Some are call positive, call negative. और उसी टाइप से ये भी होगा, ठीक है. द थर्ड वन इज रेजिस्टेंट फैक्टर, आर फैक्टर. द थर्ड वन इज आर, वेरी इंपॉर्टेंट. You know, there are some bacteria who do not die after even giving antibiotics. What are antibiotics? What are antibiotics? Antibiotics are the drugs which kills bacteria. These are the drugs which kill bacteria. So suppose there is a bacteria. There are two bacteria. This is first bacteria, this is second bacteria. I am giving same antibiotic to this also, to this also. But this bacteria are dead. After giving antibiotic, it dies. But this bacteria is not dying even after giving antibiotic. So what must I say? Why it is not dying? I will say the word resistant. This bacteria is resistant for the antibiotic. That's why it's not dying. What do you mean by resistant? What do you mean by resistant? Why it is not dying? Why it is not dying? So this bacteria, the one which is not dying, it is basically have a gene. Actually not gene, it is having a plasmid. Let me draw another plasmid. Just a second. This is the bacteria. This is the DNA of the bacteria. This is the plasmid. This is a plasmid. This plasmid have various genes for various antibiotics, like for sulfonamides, tetracycline. You know, there are numerous antibiotics, है ना? Penicillins, multiple. So whatever genes they are having, the resistant genes, if you put all those antibiotics, this bacteria will not die. If you give penicillin, it will not die. If you give sulfonamide, it will not die. If you give tetracycline, because it is having a resistant gene for them. So resistant gene, they all together known as R gene. R gene can be multiple, different for different antibiotic. So it is tetracycline के लिए tetracycline, sulfonamide के लिए sulfonamide, cephalosporin के लिए cephalosporin, likewise. So multiple R genes are present on one plasmid. Give me a thumbs up. Give me a thumbs up, everyone. So this bacteria cannot die by all these antibiotics. He was very happy. Now whether you give tetracycline, cephalosporin, penicillin, I will not die. I'm not afraid. But the friend bacteria, इसका एक friend है, दूसरा friend bacteria. उसको बहुत डर लग रहा है. This bacteria is very afraid because it doesn't have any of the plasmid for these. So if you give the same antibiotics, all these antibiotics to the second bacteria will die. So this bacteria is requesting this bacteria, can you give these genes to me also? I'm very afraid because I will die if these antibiotics will be put over me. So I do not want to die. Can you help me? You are my friend. You just replicate your plasmid and give it to me also. So they will come physical contact with each other, and this plasmid will be replicated. Donor will give it to recipient, and both will become resistant now. Give me a thumbs up if you got it. Give me a thumbs up. You got it? So let me show you the diagram. Can you see? Can you see the two bacterias? Let me show you. This is bacteria number 1. Number it 1. And this is bacteria number 2. Number it 2. Both have nucleus. This is nucleus. This is nucleus. I am not interested in nucleus. I am interested in plasmid. The first bacteria have plasmid. Can you see? Plasmid have multiple genes. One is for T for tetracycline, S for cephalosporins, sulfonamides, you know, cephalosporins, multiple. So this is R positive bacteria. And the second bacteria do not have this plasmid. So this is R negative. You got my point. You got it? Vishwas, Osama, Sambhav, and Ellipsa, you got it? So if you give these antibiotics, all these 4 to this bacteria will not die. But the second bacteria will die by these. So second is requesting the first, please do a conjugation with me, physical contact with me, and give this plasmid to me also, so that I can also not die if these antibiotics are put over me. So a sex tube is formed between them. A sex tube is formed. They do the mating, and the first bacteria will replicate the plasmid. One copy will be kept by himself, and the second copy will be given to the second bacteria. It is not the same copy, है ना? So aeroplane में पहले अपना मास्क लगाओ, फिर दूसरे की रक्षा करो, वैसा ही है. So the first copy will be kept by the same bacteria, and the second replicated copy will be given to another. You got my point? So this is the thing. This is the thing. But for transferring and replicating this, one more gene is required. That gene is known as RTF. What do you mean by RTF? It is resistance transfer factor. How can I explain you? Okay, let me draw another bacteria. This is the nucleus of the bacteria. On this is a plasmid. Let me draw a plasmid. On this plasmid, the first gene is T for tetracycline. The second is cephalosporin. The third is sulfonamide. So these 3 genes are the resistance for these 3 antibiotics. Now the 4th gene is RTF. If RTF is present, if it is present, then only the plasmid can replicate. Otherwise, it will not replicate. So RTF is very necessary for the replication. Once it will replicate, then it will transfer to another bacteria. Imagine if T, S and C genes are present, but RTF is absent. So this bacteria cannot give the plasmid to another because it cannot do the transfer. Give me a thumbs up. So RTF is must. You got my point? So on this plasmid, one RTF is present and multiple R determinants are present, different for different antibiotics. That is the point. You got it? So this is the story. You got it? I guess we are done with the chapter. We are done with the chapter. The 4 methods. Listen, you have to learn this chart. Transformation was first time discovered by Griffith in a bacteria known as Pneumococcus. ठीक है? Transduction was first time discovered in a bacteria E. coli. Lysogenic conversion was first time discovered in Clostridium diphtheria, the toxigenic and non-toxigenic strain I have told you, right? And conjugation was first time discovered in E. coli K12 strain. So you have to learn the name of the 4 bacteria in which these methods first time discovered. Give me a thumbs up. So who will tell me the summary? What are the methods? Who will tell me the summary? Help me. What are the methods of gene transfer in a bacteria? Can you tell me the 4 methods? Vishwas, can you tell me? Analipsa, Sambhav, Shubhangi, Osama, anyone? What are the 4 methods? So the first method is transformation. Very good, Analipsa. What is the second method? It's transduction. Right? What is the third method? One I have written extra. Okay, I will erase it. Okay, just a second. The third one is lysogenic conversion. Lysogenic conversion. And the last one is the conjugation. Right. Now, can you help me in defining each of them? Very good. Everyone is correct. Can you help me in defining transformation? What is transformation? Transformation is the transfer of genetic material from one bacteria to another through naked DNA. No agency is required. DNA directly coming out of one bacteria and entering in another. Why DNA is coming out of one bacteria? First bacteria is dead. Right. And Griffith experiment you have to learn. Transduction, it is through the agency of bacteriophage. The agency is bacteriophage. The agency here is bacteriophage. Yes, Sambhav, absolutely right. Right. So, bacteriophage is coming, entering the first bacteria, taking a little DNA. The same DNA is transferred to another. Only bacteriophage is acting a delivery boy, taking from one place and releasing at another place. You got my point. The third is the lysogenic conversion. Again, a bacteriophage is involved. But here, actually, the genetic material of the bacteriophage integrates with the genetic material of the bacteria. It is not a delivery boy. It is actually participating in the genetic transfer, gene transfer. And the last one is the conjugation. The word conjugation means physical contact. It's physical contact. It is of 3 types I have told you. F-plasmid, cold plasmid and resistance, R-plasmid. So one is male, one is female, one is donor, one is recipient, and a sex tube is formed, sex pilus is formed. We have learned that. Everyone give me a thumbs up. Come on, you got it? Yes, absolutely right. So you people are so smart, right? You have learned it. The 4 methods with examples, with everything. So out of the 4, lysogenic conversion is not really very important. The remaining 3 are shown here. Can you see the first diagram? Here free DNA is coming. See the naked DNA. The first bacteria is ruptured. The naked DNA is entering the second one. So this method is transformation. In the second method, a bacteriophage is involved. Can you see? The bacteriophage entering the first bacteria, taking a little bit genetic material and then infecting the second bacteria. So this method is transduction. And in the third, a sex tube is formed. From the sex tube, actually the transfer takes place, the genetic material. So this method is conjugation. So you have to learn the agency. What is the agency? Is the agency naked DNA? Is the agency bacteriophage? Or is the agency physical contact? Physical contact. So based on which the main 3 methods, again a sketch diagram, beautiful diagram, beautiful diagram. Can you see here? It's free DNA. This one is free DNA, naked DNA. So that's why transformation. Can you see this is the phage, bacteriophage. That's why it is known as transduction. And can you see a sex tube is formed? This one is conjugation. Right. Can we move ahead? Are you interested in doing some MCQs with me? Can you help me, please? So read the question and tell me the correct answer. I want to see who is first and who is fast. So, you have to correct also, right? So, all of the following are important mechanism of gene transfer in bacteria except. The question includes except. The question includes except. So, the 4 options are in front of you. Lateral gene transfer, conjugation, vertical gene transfer, horizontal. Now, I forgot to tell you about the lateral and horizontal. So, what will you say? Vertical gene transfer is the transfer of gene. Okay. Let me explain you the term vertical, lateral and horizontal. I guess you are confused. So, listen. There is a bacteria. And this is the DNA of a bacteria, right? Now, when this bacteria replicate and form the 2 daughter cell, listen the word. it is forming two daughter cell. This is daughter cell 1, this is daughter cell 2. So we know bacteria replicate by binary fission. We all know the method, the steps in the binary fission. In binary fission, we know the nucleus replicate. Nucleus double hota hai, humko pata hai. So the two daughter cell have exactly same nucleus as that of parent. Give me a thumbs up if you agree with me. So this method is known as vertical. What it is known as? You all can see it is vertically going. Vertically मतलब नीचे जा रहा है वो. Vertically, अगली generation. Basically, the word vertical means next generation. It is moving to the next generation. Automatically, the DNA is replicating and moving to the next generation to the daughter cell. This is known as vertical. ठीक है? These two are the daughter cells of the parent. This one is a parent. Parent cell. It is giving the genetic material to the daughter. This is not gene transfer. It is the binary fission. I have told you 4 methods. None of the method I told you binary fission. I told you transformation, transduction, I told you lysogenic conversion, conjugation. But I didn't told you binary fission is a method of gene transfer. It is also a method of gene transfer, but in the next generation, not in the corresponding generation. Now this parent cell, if want to give genetic material to the friend, another bacteria of same generation, it is the gene transfer. Now for doing so, 3 different methods are applicable, right? So we have transformation, we have transduction, we have conjugation. So it is in the same generation, not in the next generation. And this transfer, you all can see this transfer is in the same line because all are in same generation. So this is known as horizontal. Horizontal का मतलब है same generation, horizontal और lateral, laterally. हॉरिजॉन्टल और लेटरल का मतलब है सेम जनरेशन. यू गॉट माई पॉइंट. और वर्टिकल का मतलब है नेक्स्ट जनरेशन. गिव मी अ थम्स अप इफ यू गॉट इट. आई ट्राइड हार्ड. सो नाउ रीड द क्वेश्चन एंड टेल मी द आंसर. Now you read the question. All of the following are important methods of gene transfer except. So lateral और horizontal तो एक ही होता है, right? और conjugation is one of them, but not vertical. Vertical means next generation. So give me a thumbs up. Now all those who were wrong, kindly give me a thumbs up. You got it? So correct answer is C. ठीक है. Vertical transfer means binary fission. It is not a method of gene transfer. So correct answer is C. I guess you all got it. So can you tell me the next question? It's very easy. Drug resistance transfer by bacteriophage involves. Does it involve transduction, conjugation, transformation or convocation via the agency of bacteriophage? I'm asking the agency of the bacteriophage. Yes, the agency of the bacteriophage is transduction. You all are absolutely right. Well done. Great. So correct answer here is A. You all are right. Can you tell me the next question? Virus-mediated transfer of DNA from one cell to another. It is virus-mediated. So the agency is the virus. So agency is virus in transduction or transformation or transcription or integration. Again, an easy question. The same question actually in a different language. Yes. So in the last question I asked you bacteriophage. In this question I am asking virus. The meaning is same. So correct answer again transduction, and you all are right. So you should have ample of knowledge to solve these questions. The questions are common sense only, right? Okay, the next question, again genetic transformation by phage. So the same question with a different language. So would you go with lysogenic conversion or transduction? B or C में से क्या बोलना चाहोगे? What is the correct answer? So Priyanka is saying C, Sambhav is saying C, Annalipsa is C. So yes. So I am asking through phage, means phage is acting as a delivery boy. So I will go with transduction only. So correct answer here is C. So don't get confused with B. Correct answer is C. You all are right. You all are right. Now tell me the next question. Viral DNA actually integrated with the bacterial DNA. The virus is not acting as an agency only. It is not acting as a delivery boy. It is giving its DNA to the bacteria and both are integrating. Now what you will say? Will you say transduction or lysogenic conversion in this question? Yes. So in this question you will go with lysogenic conversion. I guess you got the concept. दोनों में देखो इस question में भी और last question में भी I am asking about the phage. I am asking about the virus. लेकिन virus delivery boy का काम कर रहा है कि खुद participate कर रहा है integration में वो that makes a sense. That makes a sense. You got my point? Yes, absolutely right. The correct answer here is lysogenic conversion. No need to explain again. ठीक है. Now tell me the mechanism of direct transfer of the free DNA, naked DNA. Naked DNA means? Yes? Yes, you all are right. transformation. Absolutely right. And the experiment is Griffith experiment, right? So I am done with it. I would like to stop here. Thank you very much for being with me. And I really enjoyed teaching you. After a long time, I came live. And from today, I will try to come live very frequently, right? So whatever topics you want from my side to be taken in the live sessions on YouTube, don't forget to text those topics on this contact number. If you have any doubt in these subjects in patho, pharma, micro, PSM, medicine, don't hesitate to ask your doubt. Whenever I will be free now, I will try to schedule those topics and I will try to reply you. So my contact number is in front of you, 9833032948. Don't forget to tell your college name, university and exam dates, right? So what you are saying, Mukul? Okay, your exams are in December. So I will try to complete the topics. Yes, malaria analysis I have already taken. You want malaria cycle or anti-malarial drugs? I mean, you want microbiology or you want pharmacology on malaria? What do you want? I am having both. So Analipsa, kindly text me personally on my WhatsApp or Telegram. I will send you the previous year sessions link, right? Previous batch link. I will try to schedule the topics which you all are requesting soon, but I get requests from, hundreds of students. Daily message me the list of the topics. So the topics which are most frequently requested, I try to schedule those topics first. But everything is in list. Don't worry. Gradually, we will move on and try to finish them as soon as possible. Now, there is a big announcement, great announcement for you people on huge demand of students throughout the country, not only throughout the country, throughout the globe, I must say. Thanks for the love by the students for me. Thank you. So I'm going to launch my own app. I'm going to launch my own app. It's MedLive. The name of the app is MedLive by Dr. Priyanka. It's MedLive. You can install the app from the Play Store. It's MedLive by Dr. Priyanka. And I'm going to launch the 3 subjects. Now, in the first phase, I'm going to launch it exclusively for second Prof University exam. So, all the candidates who are due for university exam, this is the gold standard app for you. In this, I'm going to cover the 3 subjects, the patho, pharma and micro. Patho, pharma, micro. The micro according to the new syllabus, new curriculum, right? The 3 subjects, the theory as well as practicals. Not only this, the theory I'm covering both paper 1, paper 2. So, it's like that only. Theory paper 1, theory paper 2 of the 3 subjects separately. So you are having video recordings. So just in 30 to 35 hours, each subject, I'm finishing the theory paper 1 as well as paper 2 as well as practicals. In the practicals, I'm covering all spotters, I'm covering the instruments, I'm covering all the exercises, I'm covering viva voce, I'm covering everything. So I challenge, if any student watch these hours, just 30 hours, if you spare only 30 hours, you will get gold medal in the subject. I'm challenging that. You can accept my challenge. So because each and every small, minute, long, short questions, MCQs, sample papers, previous year university exam papers, everything is covered under this in a compact, concise, beautiful manner. Not only this, I'm providing notes also. The soft copy, the hard copy. The soft copy available in the app only, but you cannot download it. You can read it in the app and make your own notes. And if you want the colored hard copy book, you can order it, right? So notes are also available. And now the end thing, what is the price of the app? Many students ask me. So it will be very nominal. Believe me, every student can afford it. It's the price is good for nothing. It's for the benefit of the students. So we are going to launch it soon. Subject by subject, we are going to launch in a phasic manner on 21st November. Wait for the date, mark the date. On 21st November, we are going to launch patho. And in the next few days only, we will launch pharma and micro also. Complete package, everything. Don't go for any book, any other book, everything is included in this only. The notes, that's it. My videos, my notes, that's it. For second Prof gold medal. So you can get 3 gold medals this year, right? So I will try to do it as soon as possible. Throughout the globe, it is applicable. From Nepal also, you can install the app, you can go for it. So it will be available all over the globe, not only in India, right? Because many students follow me from outside India also. So, right. So soon I will provide you the link. You can search it on the Play Store by the name, and soon I will, on the date of the launch, I will provide you the links also. Thank you so much for being with me, and thank you so much for your cooperation, for your love for me. And in this session, there is a link given in the comments. You can see right now, there is a pinned link. In that link, you can connect with me on WhatsApp group, on Telegram, on Instagram, on YouTube, on Facebook. I request you to follow me on multiple social media platforms so I can give you as much knowledge as I give. Daily I give 2 to 3 short videos, 1 minute learning. I have a series in which I teach a topic in a compact manner in just 1 minute. So you can secure many MCQs from those shorts. I give many MCQ practice, you know, so many study related material I give on these social media. Thank you so much for being with me. And further, if you have any request, any doubt, kindly send a personal message on WhatsApp or Telegram. But don't call, just send a message. Thank you so much. Bye-bye. Study hard. Wishing you all the best for your exams. I'm ending this session right now.